Okay, Assalamu alaikum everyone. My name is Samra Mirza and I'm a second year medical student. I hope you're all well. I'll just be covering the remainder of cell signaling. So that's part of cell signaling 2 and 3. And before I begin, these are my contact details. Feel free to message me or email me if you have any problems. And now we can begin. So what I'll be covering is the remainder of cell signaling 2, so that includes a lot of pathways, but inshallah they'll be fine. And we'll also be covering cell signaling 3, which is actually one of the more easier lectures. So I know you covered a lot of types of receptors, ions, and so on. So we're just going to be going through the remainder. So there's something called enzyme-linked receptors, and as the name suggests, they have enzymatic activity. So how that works is either the receptor itself is an enzyme or it activates an enzyme nearby. And they are single pass transmembrane proteins. What that means is they're not like the G protein coupled receptors, which has 7 loops. They just have one single loop like this. And so what they can do is when the ligand binds to them, they can either form like a dimer, which is this, and I'll get into details about what that is. And once they're bind together, they can have enzymatic activity, or once the ligand binds, they can activate a nearby enzyme. And that's what enzyme-linked receptors are. So there's two types of them. There's the tyrosine kinase receptors, and you covered them in slight detail. And then there's those which are enzymes that are not tyrosine kinase. So those are just these examples, and there's not much to know about them. You just need to know their names. If there was a question asking which one of these is an enzyme linked receptor, and so these are the options that you would have, and maybe one that's not it. So that's how you should do it. There's nothing else to know about these. And then before we move into the tyrosine kinase receptor itself, there's just some concepts that I would like to clear out in the start. So there's something called SH2. SH2 is a domain, which we'll get into detail about what a domain is, but essentially a domain is a part on a protein. So it's like a functional unit on a protein. And this one is called SH2. And it essentially stands for SRC homology 2. And again, you don't need to know what that is. You just need to know that this is a domain on a protein. And what it does is its main function is binding to phosphorylated tyrosine residues. So receptors are proteins, right? And they are made of amino acid chains. And part of it will have the tyrosine amino acid. And once this amino acid is phosphorylated, SH2 can bind to it. And that's its function. So whatever protein contains the SH2 domain, they can then bind to the tyrosine, phosphorylated tyrosine residues, and they can just like dock to it. So that's what the function of this domain is in any protein that has this domain. So proteins that contain the SH2 domain are called adapter proteins. And their examples are either the RAS protein, or GRB2, which is in the RAS pathway, or they can be the STAT protein, which is in the JAK-STAT pathway, and we'll get into that as well in a bit. But that's pretty much it for what adapter proteins are and what SH2 is. And then we'll move into tyrosine kinase receptors. So their initial structure, as I mentioned, they're just like a single transmembrane protein. So that just means if this is your cell membrane, then they're just like this. And this is your receptor. And there's 1 to 2 because they're going to form a dimer in between. Now, they contain like 3 parts, the receptor itself. So it has 2 terminals and then it has a core. So if you come here, this terminal, which has the amino group in it, it's called the N terminal because of the nitrogen. And this terminal, which has the carboxylate group in it or the COOH group, it's called the C terminal because of the C. And then everything in between is the alpha helical core. And the way I used to remember this is ligand has N in it and on N terminal, they go together. And that's how I remember that the N terminal is always the one that's facing the environment. It's outside of the cell. And the C terminal, cytosolic, it's always inside the cell. It's what's facing everything inside. And so tyrosine kinase receptors, the ligands for them are either growth factors or hormones. Moving on, we're going to break this down. So tyrosine and kinase. Kinase, whenever kinase is involved, it means phosphorylation. There's going to be phosphorylation of something. And tyrosine kinase just means phosphorylation of tyrosine. So they're going to be phosphorylating tyrosine on the receptor itself. So for tyrosine kinase receptors, they have something called, these receptors have the enzyme itself called tyrosine kinase, So if you come here, this is the tyrosine kinase, and it can essentially phosphorylate itself. So it doesn't need an external enzyme to come in and add the phosphate group. It's going to phosphorylate on its own from this enzyme. So what it does is it works like this. So this is the receptor molecule. And essentially what ends up happening is once the ligand binds to it, you can see here that this is the tyrosine kinase enzyme, and they're going to phosphorylate the opposite group. So from here to here and here to here. And you end up with the product of phosphorylated tyrosine residues, which are these. And then what ends up happening is, as I mentioned, SH2 is binding to phosphorylated tyrosine protein. So any adapter protein that contains SH2 is going to come in and it's going to bind to these tyrosine residues, and then they're going to do a signal conduction forward. But that's all you need to know for tyrosine kinase pathway. Is this much clear? I hope so.
Yes,
Anyone?
really clear.
Okay, and then this is just the same diagram from the lecture, and it's just for the sake of reference, so you have everything from your slide as well. Same concept. Then we move on to something called non-tyrosine kinase receptors. So what these mean is that there's a receptor, although it does have the tyrosine residue, it does not contain the tyrosine kinase domain. So it does not have the enzyme. So they cannot phosphorylate themselves. They need an enzyme to come in and phosphorylate them. So you'll have the cell and you'll have the receptor and you'll have the ligand. And then once the ligand binds to the receptor, there's going to attract an enzyme, and it's going to be a tyrosine kinase. And that enzyme is going to end up phosphorylating the tyrosine residues on the receptor. So examples of these are SRC, and you don't have to get into any details of them. All you need to know is SRC, if it's mutated, it's going to lead to cancer. This is all you need to know, because this can just come as a simple recall question. This is all you need to know. And then the other example of a non-tyrosine kinase receptor is JAK-STAT. So again, we're going to break down the name. JAK means Janus kinase. Again, I mentioned that they don't have the enzyme for tyrosine kinase, so they need a tyrosine kinase type of enzyme coming in. So that's the Janus kinase, JAK. And then STAT is an adapter protein, but it's also a transcription factor. So STAT stands for signal transducers and activators of transcription. So they're going to bind to the tyrosine residues, and they're going to conduct the signal forward as they tend to. And they're going to do that by acting as a transcription factor. So that's what the entire name means. And another thing to know about this receptor is that it's a cytokine receptor. So that means the ligand for it, it's going to be cytokine. So those are interferons and interleukins. And you don't need to know any examples of them. You just have to know this actually. Like this is the ligand that will bind to a JAK-STAT receptor pathway. And once the ligand binds to the receptor, there's going to be dimerization occurring. So that just means like the two receptor molecules are separated, and once the ligand joins, they come closer together and they're connected. Moving on, once they're connected, they're going to attract the JAK molecule. So they're going to attract the enzyme that's going to come in to phosphorylate the tyrosine residues. And then once they've done that, now the phosphorylated tyrosine residues are present, STAT, the adapter protein, is going to come in and it's going to dock to the receptor because it has the domain to do so. And then JAK, the enzyme again, is going to phosphorylate STAT. So now STAT is going to get phosphorylated as well. And you'll see what that does. So essentially, this is our receptor molecule, and then this is a ligand binding to it. And then because now it's dimerized, as you can see here, JAK is here, and it's going to start to phosphorylate the tyrosine residues on this. Then STAT is attracted, and they're going to bind to the tyrosine residues with their domains. And again, this is still the JAK enzyme. And what JAK is going to do, it's going to phosphorylate the STAT protein as well. So now it has its own phosphate groups. And the two STAT proteins, because there are two parts to the receptor molecule, they're going to, that's why you have two STAT proteins. These two proteins are going to dimerize together. So now it's one big molecule. And then remember that they're a transcription factor. They're going to move into the nucleus, and then they're going to carry out their effect through transcription. And that is the JAK-STAT pathway. Is that clear?
Yeah, it's clear.
Okay. And so we now have pathway done as well, we now have PI3K signal. And here, the tyrosine residues are already phosphorylated. Okay, and then we're going to move on to PI3K signaling. Now, this is something that's used in insulin signaling, and we'll come to that as well. But essentially, what occurs here is that the point that you're starting from in this pathway, the tyrosine residues on the receptors have already been phosphorylated. And so this is how we're starting off. And then this phosphorylated tyrosine residue is attracting a molecule or an enzyme called PIK3. And PIK3 is another kinase. So it's not going to be phosphorylating the tyrosine residues, it's going to be phosphorylating something else. So this is our cell membrane, and the cell membrane contains something called PIP2. And then what it does is PIK3, or the kinase, is going to phosphorylate PIP2, and it's going to convert it into PIP3. So these are the names of PIP2 and PIP3. As you can see, it gets converted from a diphosphate into a triphosphate. It's important not to confuse this reaction with the one that you see for phospholipase C, which has IP3 plus DAG. This is different. This is completely different, and this is not something that you should confuse it with. And so essentially back to it, once the kinase has converted PIP2 into PIP3, PIP3 is going to recruit a protein or an enzyme called AKT. Now, AKT is an inactivated enzyme, so once it's activated, it's going to be called protein kinase B. Protein kinase B is going to work on something called BAD, and BAD is just going to promote cell survival. So this is just pretty much all you need to know. AKT comes from the PI3K signaling pathway, and it's going to activate BAD, which is going to promote cell survival. That is it. As I mentioned, this is still a pathway, so it needs something to block it at some point. So for that, we have something called P10, and P10 stands for position and tension holding. And I don't know what that name means, but essentially, this is going to inhibit PIP3, so it's going to dephosphorylate it. So PIP3 is going to be converted back into PIP2, and therefore it's not going to activate AKT and BAD and everything. So the inhibitor for this pathway is P10. Now, this is used in insulin signaling, so we're going to come to that. So now the receptor for insulin is slightly different. It has like many parts. So it has this top part, and this is the one that's outside again. So these are the alpha chains. So this is both alpha chains, this and this. And then it has the inside part, which is the beta chains. And so there's again two of them here. So there's a total of 4 subunits, again, 2 for each part of the receptor. It also has an SH2 domain present on itself, so it doesn't need an adapter protein anywhere near. And what you need to know about insulin signaling is that this is a tyrosine kinase receptor, so it does have like inherent tyrosine kinase ability. It's not like the JAK-STAT pathway. So what ends up happening is once insulin binds to the receptor, what's it called? The tyrosine kinase activity of the receptor itself is again gonna autophosphorylate. So they're gonna have the phosphate groups, and then it's going to attract IRS, which is insulin receptor substrate. Now, this molecule can like act on different pathways. It can act on or like start different pathways. It can start the RAS pathway or the JAK-STAT pathway. But the one that is important for insulin signaling is PI3K. So that's just the one we did here. And it's this exact same thing, and it's going to start it. And what AKT is going to do is AKT is going to allow for GLUT, which is a receptor for glucose. It's going to allow it to move from inside the cell to outside. So it's going to, like, the receptor is going to become part of the cell membrane instead of just being inside. And then glucose can now finally move into the cell, and this allows for the actions of insulin to take place. You need to know that this is for GLUT4 and not the other types of GLUT. And that is it for insulin signaling. These are the biological effects of insulin. So again, what essentially it's doing is insulin again just removes anything that's like giving you energy. So it's gonna allow glucose to go into the body instead of being inside your blood. It's gonna allow glycogen synthesis, so it's storing the glucose. It's gonna cause protein synthesis, so it's storing more protein, and it's gonna cause fat synthesis. So it's everything is in store mode instead of using mode. and it's going to inhibit anything which is doing the opposite. And then, is this part so far clear for insulin signaling?
Yeah, it's clear.
Okay, then we're going to come into some clinical stuff for this, and this slide is pretty ugly, so we're going to come here. Um, Insulin, okay, diabetes. Diabetes has two types, and I'm sure you all know this, but there's type 1 and type 2. The one of importance in this case is type 2, which is where the body is producing insulin normally, but the effects of it are still not coming through. So if it's being produced properly, then that means that the problem has to be with the receptor or the pathway, somewhere in between. So the problem is in the tyrosine kinase receptor pathway. So instead of tyrosine getting phosphorylated, which is usually what happens, serine gets phosphorylated instead. So the problem happens right in the receptor, and then it cannot like you cannot get the GLUT4 to come out. You can't even get the PI3K pathway to start because from the get-go, it was tyrosine was replaced by serine phosphorylation instead. So this is done by an enzyme called SOX3. And SOX3 is, you'll see it here actually. This is SOX3 and it's an enzyme. And instead of like PI3K in that entire pathway continuing, this pathway started from SOX3. And SOX3 is stimulated by these two factors, interleukin 6 or tumor necrosis factor alpha. You don't need to know them in detail. This is mostly for the sake of understanding. These two factors are normally increased when a person has inflammation. And because type 2 diabetes is associated mostly with obesity, in obesity, they also notice that a person just generally has more inflammation, and these factors are just normally more increased in a patient with type 2 diabetes or obesity. So because of an increase in these two factors, SOX3 is going to get stimulated and it's going to cause serine phosphorylation instead of tyrosine phosphorylation. And then that stops the main pathway from occurring. Another thing to note is that tumor necrosis factor alpha also has its own pathway, and it's this one. Again, you don't need to know them in detail, but if you do see this name, PKC beta, and you see a tumor necrosis factor alpha, it's for type 2 diabetes, insulin resistance. So this is one pathway, which is again tumor necrosis factor alpha acting with PKC beta. You don't need to know the details. Or it's interleukin 6 and tumor necrosis factor alpha acting on SOX3 and stimulating serine phosphorylation instead of tyrosine. Those are the key points of this. And then again, just for the sake of your information, I'm sure you all know this. In type 2 diabetes, there's insulin resistance. So the receptor is not acting properly to the insulin, even though it's still binding normally, it's just not acting normally. And because the effects of insulin are not coming through, there's going to be hyperinsulinemia in the start. And eventually the pancreas are going to get tired and fatigued, and there's going to be a decrease in insulin levels. And so type 2 diabetic patients end up becoming insulin dependent as well. And what's it called? For insulin, so natural insulin, when it's produced in your body, it's produced with C-peptide. And this is just a protein that's like onto the insulin as well. So if you want to measure whether insulin is being produced in the body or not, you usually look at C-peptide. And so for people who are insulin dependent, they would have a low C-peptide level. And with that, we conclude, I think, all of cell signaling too. Is everything clear so far?
Yeah, everything is really clear. Thank you.
Okay, so we have some questions. So you can just um let me know when you want me to move forward. I'll move on to the next slide now. That's okay. And so this is the answer. Is the answer clear?
Yeah, it's clear.
Yeah, so again, insulin signaling is just, it's related only to this pathway. None of the other pathways are at all linked to insulin signaling. This is again from the PI3K pathway. And this is the next question.
Would it be A?
Okay, why do you think A?
No,
Look,
first of all, I kind of don't understand the question as in impaired response manifests as an inability to recruit the insulin response substrate 1 protein to the receptor.
mm-hmm.
Hold on. I just don't understand how the question was worded.
Okay, I'll explain this actually. So what's happening is they noticed that one person is responding normally to insulin while the other isn't. And they noticed that in both, like the receptor is binding normally. Like that's one thing, right? They have equal affinity. So there's no problem with the binding itself. But the impaired response manifested as an inability to recruit the insulin response subject, so the IRS1 protein, to the receptor. So if we go back to the insulin signaling diagram here. Remember that once the ligand binds to insulin, it has these tyrosine, like it gets the tyrosine residues autophosphorylated through its integral activity, and then it recruits IRS, right? And then IRS is the one that ends up starting the PI3K pathway. So they're saying that in this question, they noticed that right, they're saying in this question they noticed that the IRS protein isn't getting recruited at all. So they're saying, what is the problem here? So RAS-G isn't included in this entire insulin signaling pathway, right? And then they're saying tyrosine phosphorylation of the insulin receptor leading to loss of IRS binding site. But if tyrosine phosphorylation is present, then why wouldn't IRS bind? So that's why B is wrong. C gamma, this whatever, isn't related again. PI3 kinase phosphorylation of insulin preventing IRS binding. Again, this isn't applied just yet because PI3 kinase starts after IRS. Like once IRS is there, that's when PI3 kinase is going to start its work. But so the problem is that IRS itself isn't coming to the receptor or not binding to it. So the problem is mutation in tyrosine phosphorylation site. Like the tyrosine phosphorylation site has to, like the tyrosine phosphorylated residues have to be there, and they're the one attracting insulin response substrate. So if they're not present or if there's a problem in that thing itself, like if it's abnormal from its original, I guess, position or anything like that, that's when IRS wouldn't be recruited, and that's why the pathway wouldn't start. Does this make sense?
Yeah, it does make sense.
Okay, good. Alhamdulillah. Okay, would you like a little break before cell signaling, or would you like to start immediately?
I mean, I don't mind.
Okay,
Uh, okay. If I mean, I guess we can continue.
okay, it's very short, inshallah. I'll go by quickly. So we'll move on to cell signaling 3, and this is very, very easy. So I'm pretty sure you know this from Endorepo, but again, hormones are they're chemical messengers as opposed to neurotransmitters, which are electrical messengers. And so hormones are chemical messengers, and they're again just for cell signaling and cell communication. They're transporting signals. There's four main types of hormones. So you have steroidal hormones, you have peptidal hormones, amino acid derivatives, and prostaglandins. So for steroidal hormones, you learned this recently. There's glucocorticoids, mineralocorticoids, and gonadosteroids. So if there's just a question asking, this is a hormone, what type does it come under? You can obviously, inshallah, be able to choose them from steroidal at least. And then peptides, insulin is a protein. So as a hormone, it's a protein hormone. So that's why it's a peptide. As for amino acid derivatives, epinephrine and norepinephrine are both examples of amino acid derivatives. And PGE2 and other classes of prostaglandins, I don't think you guys have taken them, but just PGE2, they come under prostaglandins. Another thing to note about steroid hormones, like you all obviously, hopefully, should know this from Repro, but they affect inflammatory responses. They have cardiovascular functions, bone metabolisms, and so on and so forth. This is just in general details about them. Examples of the steroidal hormones, there's glucocorticoids, and under them you have the cortisol. For mineralocorticoids, you have aldosterone, and for gonadosteroids, you have androgens. So that comes under and then that has testosterone, and then you have estrogen and progesterone, and so on. And then we move into nuclear receptors. So these are, as the name suggests, the receptors that are on the nucleus. So they have a specific structure and function to them because for steroidal hormones, they mostly act intracellularly, and we'll get into that. And that's why the nucleus has the receptors. But for these receptors, their general structure, right? These are the structures and these are their functions. So they have a domain, and this is a variable domain. So this part is what interacts with transcription factors. So what you'll notice is that steroid hormones, their general mode of acting is by in like affecting transcription and then affecting gene expression and so on. So they mostly act through transcription. That's why there's a domain that is for interacting with transcription factors on the receptor itself. Then the receptor has another domain called DNA binding. So what happens is, we'll get into this mechanism again, but the receptor, once the ligand binds to it, which is like the steroidal hormone, for example, this receptor is going to move into the nucleus. It's going to go inside just the surface. And once it goes inside, it's going to bind to the DNA in certain parts of it. And to do that, you need to have a DNA binding domain. And for this, they use something called a zinc finger motif. And we'll get into the difference between a motif and a domain, but that's what they use. So the DNA binding domain is made of like zinc finger motifs, and they are to recognize which part of the DNA they want to bind to, so like a specific sequence. Then the receptor has a domain for dimerization. You don't need to know this in too much detail, just like when there's two receptors and they need to dimerize and become like one main thing, there's a site for that. And that's again on the nuclear receptor. It has a specific site for each of these. And then there's a hormone binding domain, which is the actual ligand binding site. So that's where the ligand actually has to bind itself. So again, there's four main parts to the receptor. There's the variable domain, which is for transcription factors. There's the DNA binding domain, which, as the name suggests, binds to the DNA and it uses zinc finger motifs. There's the domain for dimerization, which is when the two receptors come together. And then there's a hormone binding domain, or the actual binding site. And then we move into motifs versus domains. Now, they're both part of the protein, which is the receptor, what the receptor generally is. The difference between them is, first of all, a domain is much larger and a motif is much smaller. An example of a domain was the kinase domain, like the tyrosine kinase domain, and it's a protein and it's an independent structure from the actual like receptor itself. Like the tyrosine kinase receptor was there, and then it had the tyrosine kinase domain. Now the domain isn't affecting the receptor action of it, it's affecting the signal transduction action of it. Like its function is separate from the main thing itself. Like it has its own specific function, like a domain does. Rather than a motif, like the function of the motif is to like help with the original like structure too. So like you notice zinc finger motifs, they're not doing something additional, they're just helping in binding and so on and so forth. So the nuclear receptor has to bind to certain places, and that's what this zinc finger motif was doing. So the difference is the motif is its role is associated with the main structure it's on. As for the domain has its own function. It's separate. That makes sense. A motif is smaller and the domain is larger. A motif becomes more complex. It has like multiple secondary structures, like it's at a higher level of protein development. And so certain examples that you should know are helix, turn helix, zinc finger, and ATP binding motifs. These are just good examples to know. As for domain, the only one you need to know is the kinase domain. Is that difference clear?
Yes.
Okay, good. Then we move on to steroid receptors. Now, there's two types of steroid receptors. They can either be on the cell membrane itself, like this, or they can be inside. So if this is our ugly cell, and this is one receptor here, or the receptor here. So the receptor can be a membrane receptor, or they can be inside the cell. So these two receptors. And what they can do is either they can be free-flowing in the cytoplasm, or they can be attached to the nucleus. And then this essentially is just an explanation again of steroidal hormones. Because they're made from cholesterol, which is mostly hydrophobic, they can pass through the cell membrane easily because they have the same hydrophobic structure, but for passing through the bloodstream, which is more hydrophilic, they need a transport protein. It's just some basic understanding. I'm sure everyone knows this. Then for the intracellular receptors, as I mentioned, there's either one that's free-flowing in the cytoplasm or there's one in the nucleus. As for the cytoplasm one, that's type 1. And you need to know this distinction that type 1 is for cytoplasm and type 2 is for the nucleus. Type 1 cytoplasm works with heat shock proteins. Again, just a characteristic feature, no details here. This is the very, very important part, which is examples of these receptors. So sex hormone receptors, androgen, testosterone, estrogen, whatever, so on and so forth. Those are under type 1 receptors. Glucocorticoid receptors and mineralocorticoid receptors, sorry, they're also under type 1. And those are just examples of them. The way they work is essentially once the hormone binds to the receptor, they move, they're intracellularly, they're in the cytoplasm, the receptor and the hormone bind, and then they move in. They move into the nucleus and they bind to the DNA. And because again, their process of steroid hormones in general is through transcription. So they bind to the DNA through a structure called hormone response element, and they move in as something called a homodimer. A homodimer just means that a dimer, which is like made of two molecules together, homo means same. So like the dimer is composed of two identical molecules. Like it's not like one part will be completely different and one part will be completely different. And I'll show you an example of that. But essentially just know type 1 hormone and receptor bind, and then they move into the nucleus, and then they have to bind to the DNA. They bind to the DNA through the hormone response element, and they do this as homodimers in type 1, and then they affect transcription. That is it for type 1. As for type 2, they're in the nucleus, so they're inside and they're bound to the DNA. So they can either be on the surface of the nucleus or they can be inside it, and they're essentially bound to the DNA. And again, the steroid hormones can easily pass through. Examples of these are the thyroid hormone receptors, the receptors for vitamin A and D, and retinoid receptors. There is no way to not know these examples. There's no shortcut. You should know these by heart. Now, they also form dimers, but they can form homo and heterodimers. So what that means is, this is an example of a homodimer. So you can see that it's made of the same things. This is an example of a heterodimer. So there's RXR on one and RER on the other. You don't need to know what these two mean. It's just an example to show that this is what hetero looks like and this is what homo looks like. Back to it. So this receptor, the type 2 receptor that's in the nucleus, when it's not bound to a hormone, so in a normal state, this receptor is linked to something called a co-repressor protein. And this co-repressor protein prevents it from doing transcription of certain genes because, again, each receptor and its ligand has a specific function and they carry this out through a specific part of the DNA. So in normal conditions, when this hormone isn't released and its action isn't needed, this transcription of certain genes has to be prevented. So that's being done through the co-repressor protein. Once the hormone binds to the receptor, the co-repressor protein is released. And now the receptor and the hormone can work together to initiate transcription of the gene that's related to their function. An example that came under type 2 was estrogen receptor alpha, which exists mostly in the nucleus. Estrogen receptors can also exist in the nucleus and the cytoplasm, but estrogen receptor alpha exists mostly in the nucleus. Does that make sense?
Yeah, those.
Okay, hopefully. Good. Then we just come into the very simple pathway for steroid hormones. So we'll start here. Travel in the bloodstream. They need the a, a like substance carrying it through. Once the steroid hormone has reached the cell, without any receptor, anything else before this. Okay. So the process of how steroid hormones carry out their action is through this. Wait 1 second. Okay, then we come on to the pathway of how steroid hormones work. So we'll start from here. The steroid hormones are hydrophobic substances, so to be passed through the blood, they need to be attached to something hydrophilic. So they have a protein carrier here. Once they reach the cell membrane or of their destination cell, they detach from the protein and then they can move in either freely or they can choose to attach to a cell surface receptor, like a membrane receptor. So we'll come to this pathway first. They can either bind to a cytoplasmic receptor, which is, as I mentioned, a type 1 receptor, or they can move into the nucleus itself and they can bind to a type 2 receptor. Either way, what they're going to do is once they have bound to their respective receptors, either in the cytoplasm or in the nucleus, they're going to bind to DNA and they're going to affect transcription and they're going to allow for certain proteins to be produced to carry out their action. Now, this takes time because it has to bind to the receptors, it has to unravel certain parts of the DNA and allow transcription to occur, then translation, and then protein synthesis. This is a lengthy process. Therefore, this pathway is long, and we're going to come into what it's called. Or it can bind straight to a membrane receptor. And when something binds to a membrane receptor, it has the whole signaling cascade of secondary messengers and enzymes and so on. And this one is usually faster. So this is a rapid response. That's the basic understanding of their pathways. Then we go into certain more details. So it's the same idea generally. We have something called classical signaling and we have something called rapid signaling. For classical signaling, it's something called as NIS, so nuclear initiated steroid signaling, and as the name suggests, it's occurring in the nucleus, and that's what we just learned, the basic pathway. They bind to an intracellular receptor and they go in and they interact with the DNA and they have protein synthesis occurring, and this takes time. It can take hours or days, so it's long. And usually what happens in this pathway, this is something from your slides, and all you need to know is when the hormone isn't present, so the receptors are there and they're not bound to any hormones, this is their locations. So estrogen and progesterone receptors are in the nucleus. Type 2 receptors are also in the nucleus, and then glucocorticoid and androgen receptors are in the cytosol. This is just from their general locations. Once the hormone binds to them, they can they can move into the nucleus if they're not already in the nucleus, and then they can bind to the DNA via the hormone response element and allow transcription to take place. The same basic pathway, just recap. As for rapid signaling, this is called as MIS, so it's membrane-initiated steroid signaling, and this is the one I showed here, which is the rapid response. So what ends up happening here is they bind either to an intracellular or mostly membrane receptor, and then they increase the secondary messengers. So protein kinase A, protein kinase C, phospholipase C, and RAS, and they increase their activity. And then they also increase intracellular calcium levels. This is usually like, again, another recap question, which like signaling is associated with increasing calcium levels. So that is rapid signaling. And because this one, well, is rapid, it has like a latency of seconds. It can be done very, very fast because it's not undergoing protein synthesis and all that. It's just enzymes and like ions and so on. So that's the basic pathways and their signaling. Then we come into like second last slide in terms of like information, and then you have questions. So for the receptors and their specificity, all you need to know is when a hormone binds to the receptor, it's going to undergo some changes so that can fit it perfectly. So the receptor is going to have some under like conformational changes. And when it's having these changes, as I mentioned, it has a DNA binding domain and that has a zinc finger motif. So after the changes occur, the zinc finger motif is going to be revealed. And then it's revealed, and now this hormone receptor complex can go and bind to the DNA because the zinc finger is available now. It's going to bind to the specific DNA sequence, and then they're going to allow for transcription to take place. To bind to the DNA, they again need a hormone response element, as we've mentioned many times. And an example of a hormone response element is GRE, which is glucocorticoid receptor element. And I hope that's clear. And then we have the last slide, which is just comparing the latencies or the time it takes for all of the cell signaling methods we've taken. So, oh wait. So for ion channel, so these are just the normal receptors or the channels that you have. Their action occurs in milliseconds. So they're like the fastest. And this has been a question in our exams, which one is the fastest? So those are ion channels. Then you have the GPCRs and the JAK-STAT and the membrane receptors, and their time is usually seconds and minutes. Then you have the steroid receptors, which when they're in their classical signaling phase, they occur through hours or days. Is everything clear so far?
Yeah, everything is really clear.
Okay, perfect. So then we can move on to some questions. Um, okay, questions.
Um, okay, so I think it would be B.
Yep, it's B. Very correct. Because again, AKT is just allowing the glue channels to move into the cell membrane itself and allow glucose to move in. It wouldn't induce gluconeogenesis. That's nowhere near in the insulin pathway. It wouldn't decrease glycogen synthesis either, and none of these are related either because PI3K occurs before AKT comes in. And then this is the next question.
Does it bind to specific DNA sequences?
Yes, absolutely. Perfect. And the rest of these are just membrane pathways. So they're not related to any of this. Next question.
Ion channel receptors.
Facts. That's it. Here, sorry. Damn it. I hope you didn't see the answers. Okay, next question.
Right, how do steroid hormones produce fluorescent cells? I think they bind to intracellular receptors and promoting transcription.
Yep, perfect. And this is just not, they don't directly inhibit protein synthesis. They don't have that effect at all. And then these are also not related because their main pathway is starting from the transcription, not from anything else. And then this question. This is
Uh,
the last question.
the sarcomology 2 domain is protein domain that recognizes bind to which I think it's tyrosine residues.
Yes, perfect. So A. These are obviously something you shouldn't get confused with, and they react with phosphorylated and non-phosphorylated. That's why this is wrong. This is obviously wrong, and serine residues don't come in at this. With that, we are done with this pal. Thank you so much, and I hope this helped. And if you have any questions, feel free to contact me or Samhar at any point.